Hi, this is Mr. Yeoman, and we are trying to continue our conversation about how DNA ends up converting into the things that we see in living things. So today what I want to do is go through some information about what you already know about how DNA behaves, and then hopefully do a little bit of application of how this translates into the physical appearances of people, or plants or animals, any living thing. So I have a sheet here for you, which is talking about from gene to polypeptide, and it's just talking about some of the rules for base pairing that we've already done. And just as a little bit of a review, we're going to go through and kind of fill out this flow chart. Not essentially a flow chart, but to kind of fill in and look at how do we go from the genetic information to the characteristics or traits of a living thing. Okay, and the only things that we're putting into these blanks are DNA, protein, and RNA. So a nucleotide sequence in the blank of a gene. Well, genes are the things that we know of in genetic information, and we know that our genetic information is made out of DNA. So genes, on a single strand of DNA, we end up having lots and lots of genes on there, which each code for a different protein. Right? So we did an activity last week where we took that DNA gene and we transcribed it, we rewrote it. So we know that the DNA can't leave the nucleus, so we have to transmit that information in a different way. So the nucleotide sequence in messenger RNA, remember there are different types of RNA, and messenger RNA is one of those. It transcribes, it rewrites the DNA, and then it takes that information out of the nucleus. Okay, so it's not taking the actual DNA, it's taking the information in the form of messenger RNA somewhere else in the cell. So now we have amino acid sequence and a polypeptide, which folds into a blank. So we have the DNA in the nucleus. The DNA is being copied and transcribed into messenger RNA. That messenger RNA leaves the nucleus, where it ends up getting pinched between two pieces of ribosomal RNA. So it's going into the ribosome. And it is going to take those amino acids. Again, we saw that video last week, and we did an activity where we were actually building those amino acids. And those amino acid chains fold up to make a molecule called a protein. And hopefully you recognize that from earlier in the year. We know that proteins are made out of amino acids, so that would make sense that if we are making a long chain of amino acids, that would be a protein. So the structure and function of the protein is made from, or is determined by the order of those amino acids as well as how they fold. So there's really four different levels that make the protein behave the way that it does. But that first level is really what are the amino acids that it's being made out of. And so depending on what those amino acids are, that's going to affect the structure and function of that protein. So that is the difference between normal hemoglobin, which carries oxygen, and sickle cell hemoglobin, which does not carry oxygen as well. It actually clumps up and causes some very serious problems for people with sickle cell anemia. And so that ends up resulting in a person's characteristics or traits. So something like sickle cell anemia you may not be able to see by looking at someone, but there are plenty of other traits that we do typically recognize. So hair color, skin color, these types of characteristics we notice all the time. But that is not the only type of characteristics. Most of the characteristics that we're talking about are things that go unseen. So looking at how do your kidney cells function, how do your lung cells function, how much mucus is produced, like in the case of cystic fibrosis. So there's all these different characteristics and traits that are being determined based on the amino acids making that protein, and that protein is made from the messenger RNA, and that messenger RNA is determined by the DNA that is there. And so there's this whole series of steps that we're going through. So again, a little bit of a review from last week. We know that we have this double helix structure, so we have our DNA molecule here. It's made out of all of these different bases that go down the center. We know that this is just a sugar phosphate backbone. It's really just holding these bases in place so they're not floating all over. So we need to know how they end up copying. So we have to have these base pairing rules, and one of the things that we were talking about last week is trying to find the other side of that DNA strand. Since DNA is double-stranded, we need to be able to find that other side. So opposite of cytosine is guanine, so we're just going to put a little G in there. And opposite of guanine is cytosine, because they're a base pair. If you have one, the other one is present as well. The opposite of adenine is thymine, and the opposite of thymine is adenine. So we have these base pairs. We know they're opposite of one another. Now, that mostly works for RNA. However, as it says in the paragraph above here, RNA doesn't have thymine. So instead, it is uracil that is in RNA. So that should be pretty easy because we still have guanine. That's opposite of cytosine. We still have cytosine. So those are still a base pair. The only difference is now we have uracil opposite of adenine. And opposite of thymine, well, adenosine is still there, or adenine is still there. So we are only changing one base. So the base pairs do change slightly when we're talking about RNA. Okay, so now we're talking about making that transfer RNA. So thinking back to that activity that we did, we had those yellow pieces that were attached to the amino acids in that video. You had those very large structures, and there was just that little red tip on there. That little red tip was the amino acid, and so that was being brought over. But we need to have that anticodon to read the codon from the messenger RNA. So since messenger RNA and transfer RNA are both RNA molecules, we just still need to go through and look at what their complementary bases are. So again, cytosine and guanine, guanine and cytosine, adenine and uracil, and then this time, because the messenger RNA is already made out of uracil, it's just looking very similar to what we had up here, where we're just simply replacing thymine. Okay. So looking at this, the first row of the table gives the nucleotide sequence and the coding strand of a hypothetical mini gene. Okay, so most of the time these genes are very, very long. You're talking at a minimum, hundreds of base pairs. And, if you're looking at an entire strand, you're talking about an entire genome being billions of base pairs. So this is a very, very short segment. But we do the same thing that we looked at up here. So we have one strand of DNA, and looking at the other strand of DNA is just going to be what is paired with it. So opposite of guanine is going to be cytosine. Opposite of adenine is thymine. We have another cytosine, guanine, thymine, adenine. So that's great for our other strand of DNA, but now we're going to transcribe that into messenger RNA. So we have our guanine here. Opposite of thymine is going to be adenine. Opposite of cytosine is going to be guanine. Opposite of guanine is cytosine. Opposite of thymine is adenine. And opposite of adenine, this was the one that's going to be a little bit different because, again, DNA has thymine, but messenger RNA does not. It has uracil. So we now have a messenger RNA that is GAGCAU. Okay, so now that we have all of our messenger RNA, we can write what those codons are going to be. Okay, so we're just breaking them up into groups of 3. Sorry, my pen went out there a little bit. So we have GAG as one codon and CAU as another codon. Now, this is important because now that we have our transfer RNA, we need the anticodon that goes with it so that we get the appropriate amino acid to go in there and make the right protein. So again, we're looking at RNA. It's just a different type of RNA, so we're looking at those same 4 bases. So we're going to be looking at C, U, C this time because opposite of adenine is uracil. There's no thymine. And G, U, A. Okay, so pretty simple. That should be all review from what we did last week. Which strand of DNA has the same nucleotide sequence as messenger RNA? Looking at that messenger RNA, it is basically the same as that opposite strand of DNA. We just changed that base a little bit. We're trying to make our different amino acids. And so that's where this is going to come in. I'm going to skip down here a little bit, because what I have here is a codon chart, and some of you may have seen this last week. But this helps you determine what base is going to be made. So if I look at this front side, my codon chart, they are typically looking at messenger RNA, and it says it right there at the top, it's messenger RNA. And so looking at this, if I find my messenger RNA, one of my codons is GAG. So that tells me that if I come over to my codon chart, my first base is G, my second base is A, so that means I'm going to be somewhere in this box, and then my last base is G. So I'm going to be making glutamic acid for that particular amino acid. Okay, we could go to the next one, and we would have CAU as our messenger RNA. And so we started with glutamic acid, now CAU, we have CA, so we're going to be in this box somewhere, and then U means that we're going to produce histidine. So we have our first two amino acids, we're calling it a mini gene, because there really isn't anything that is made out of two amino acids. But using that, you should be able to determine what those links are. Okay, what I'm going to ask you to do is, I have a separate activity for you, and you are going to be figuring out a mystery organism. And based on what amino acids are produced, you can determine what traits that particular organism is going to have. Now, this is kind of oversimplified of how a real organism works, but the concept is the same. Depending on what the order of your amino acids are, that may end up changing what that trait is. And we're going to end up going and talking about mutations and some of those items. And so what I'm really looking for you to do is get through the back of this front page, and I just want you to kind of look at what those pieces are, determine what your messenger RNA is, use your codon chart to determine what amino acids are going to be here, and then that way you can figure out what is that trait going to be. So you have your DNA, you can determine the messenger RNA. From the messenger RNA, you can find out what amino acids are being produced, and then from the amino acids that are being produced, you can look at this front chart and determine the trait that is being made for that organism. Okay? So that's what I'm asking you to do today. If you have questions, please hold on to them, and I'll talk to you about it when I get back. Otherwise, I have every confidence that you'll do great with this, and we will talk on Tuesday. Have a good day.